So chrysophytes, they are microscopic. So what is meant by microscopic? They are not visible to the naked eye. They are very micro. So diatoms, they are very beautiful because they occur in different structures. Why? Because they have calcareous shells. So slime motes are saprophytic, means they usually feed on the dead organic matter. Hello everyone, a warm welcome to today's session on chapter 2, that is biological classification. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, under the kingdom Monera, which was one of Whittaker's system of classification, we had studied about bacteria. Under bacteria, again they were classified as eubacteria and archaebacteria. So in today's session, we shall talk about the second kingdom classified by Whittaker, that is Protista. So kingdom Protista, they are eukaryotes. Means they have, the organisms have a true nucleus. That is, it is surrounded by a nuclear membrane. So they are single-celled, that is unicellular, but in spite of being unicellular, since they have a true nucleus, that is the genetic material is covered by a nuclear membrane or it is surrounded by a nuclear membrane, and that is why they have grouped them under eukaryotes. Therefore, instead of putting, now you can imagine why didn't they put Protista under the kingdom Monera, even though they are unicellular. Why? Because in kingdom Monera, the bacteria, they do not have a true nucleus, wherein they do not have a nuclear membrane. But in spite of them being unicellular, but here kingdom Protista, though they are unicellular, they have a nuclear membrane there. That is why they are put under the eukaryotes. And next is, they can be aquatic. What is the habitat? They are aquatic, means they live in waterlogged condition. They live in the water. Therefore, their habitat is aquatic. And the cell body of members of Protista, they contain a well-defined nucleus. Well-defined nucleus means the nucleus is covered by a nuclear membrane, and that is why I told you they are eukaryotes and therefore they have been put. So you can now again ask me, they are eukaryotes. Why didn't they put them under Plantae or Animalia? Why? Because they are unicellular. But whereas Plantae and Animalia, they are multicellular. That is why it is grouped separately under the kingdom Protista. And they have other membrane-bound organelles, that is the mitochondria, the Golgi bodies, all these organelles also are present. That is why they are put under a separate group. These organisms are put under a separate group called as Protista. And these organisms have flagella or cilia. So what is flagella? Flagella is a whip-like structure. So if you have an organism like this, they will have a whip-like structure. And they can also have cilia. So the difference between flagella and cilia is, flagella, it is a long, slender, whip-like structure, whereas cilia, they are small hairs. So the body is covered by small hairs, minute hairs like this. That is why this is cilia, whereas this is flagella. So some of the organisms under Protista, they may have a flagella or they may have cilia. So both these flagella and cilia make them motile. That is, they help in the movement of the organism from one place to another place. And their reproduction, that is the Protista, they reproduce asexually by fusion. That is, fusion is two cells join and they reproduce. They come in contact and they reproduce. So they reproduce asexually by cell fusion, and also they can reproduce sexually by zygote formation. Now you can ask me, why can't they put them under Animalia? Because they reproduce sexually by zygote formation. But again, animals, they are multicellular, whereas these organisms, they are unicellular. That is why it is put under Protista. So you can see here, this is Protista and you can see the flagella. So these flagella, they help in the movement of the organism. So either they may have a flagella or they may have hair-like covering like this. Some organisms may have hair-like covering like this, which is nothing but cilia. That is the structures that help in the locomotion or the movement of the organism. So we shall study in detail the different types of organisms under Protista. So the first one, it is called as Chrysophytes. So Chrysophytes, they are microscopic. So what is meant by microscopic? They are not visible to the naked eye. They are very micro. They are very, very minute. They are not visible to the naked eye, which means you cannot see them as such. Only you can see them under the microscope. So what can be done to observe them under the microscope? Simple. You can bring a pond water in a beaker or in a glass and put a small drop on the slide and observe it under the microscope. Why water? Because I told you, Protista, these members, they are aquatic. They live in water. So any pond water, if you collect and observe it under the microscope, you can see this Chrysophytes. So Chrysophytes, they are microscopic and they float passively in water current. That is why they are called as planktons. Planktons are those organisms that float in the water current, along with the water current. So Chrysophytes are nothing but planktons which are microscopic and they float passively in water, and they can be either fresh water and they can also live in salt water or marine or oceans, that is oceans or seas. So they can live in fresh water such as ponds, rivers, lakes, etc. and they can also live in marine waters such as oceans and the seas. And they are photosynthetic. So now you can tell me, they are green in color, they are capable of synthesizing their own food. Why did they not put it under? So what Linnaeus thought was, so now Linnaeus put this under plantae. Why? Because they are green in color, they are photosynthetic, that is why. But he didn't study about the cell. This is unicellular, plants are multicellular. That thinking he didn't have. But what did Whittaker do? He put, though they are photosynthetic and they appear green, he put it under a different group, Protista. Why? Because they are unicellular, they are single cell, that is why. And they are photosynthetic. The best example for Chrysophytes is Diatoms and the golden algae, that is Desmids. So you can see Desmids and Diatoms here. So talking about Diatoms. So diatoms, they are very beautiful because they occur in different structures. Why? Because they have calcareous shells. Like if you see the flower pollen grains, right? Pollen grains also, they are very beautiful structures. Different varieties of designs of pollen grains you can find because they have a exine layer which is very beautiful, decorative. Just like that, these diatoms, they have these calcareous depositions on them, and these calcareous depositions give beautiful shapes and structures to the diatoms. You can see here how beautiful, it's like as if somebody has carved on them. It is very beautiful to look under the microscope. So diatoms, they are the chief producers in the oceans because most of the organisms that are present in the ocean, they feed on this diatoms, and they have a very thin cell wall. And these cell walls, they open to make a flap-like structure. They have very thin cell walls. So these cell walls form two thin overlapping shells. And also the walls are embedded with silica. So this silica is indestructible. That is why diatoms can live on this earth for millions of years as such. There is no death for the diatom. Why? Because the silica that is present on the diatom, it gives a long shelf life to the diatom, and also it is indestructible. That is, you cannot destroy it. It is very rigid. It is very hard. And the walls are embedded with silica, and thus the walls are indestructible. You cannot destroy the walls because the walls are very, very hard, made up of silica. And there is one more term called diatomaceous earth. So since diatom, it is covered by silica which cannot be destroyed, millions of years, these silica got deposited in the earth, in the oceans. So therefore, it is called as the diatomaceous earth. Why? Because it is indestructible. Since it cannot be destroyed, they got collected millions of years, and even till today they are collecting, they are accumulating in the ocean beds, and that is why the present earth is called as diatomaceous earth, wherein it is filled, the soil is filled with silica. Why are the ocean beds filled with silica? It is because of, or why are the river beds filled with silica? It is because of the diatom, and that particular term is called as diatomaceous earth. And this diatomaceous earth, the soil of the diatomaceous earth, which is rich in silica, it is used for polishing. So polishing certain articrafts and all that, it is being used. It is used for polishing, and it is used for filtration of oils. Since they are porous and since they have the capacity to filter or to refine oil, they are used for filtering oil and also for filtering of the syrup. So when you filter, when you put it in a silica filtrate, you might have seen filters that we have at home. There is one candle that is being present, or we have filtrates which filters the waste matter. That is nothing but made up of silica, because it has the capability to filter the debris and the waste material. So therefore, it can be used for filtration of oils and syrups. So this is about the diatoms. So under kingdom Protista, we studied about Chrysophytes. So after Chrysophytes, we shall study about Dinoflagellates. So Dinoflagellates, obviously the name itself suggests di means two, flagellate, they have flagella. These organisms, they have two flagella. But earlier, the Chrysophytes, they had one single flagella, or they were made up of cilia. But Dinoflagellates, they have two flagella. So these dinoflagellates, they are marine in habitat, that is, they are usually found in oceans and seas. They are marine in habitat and they are autotrophs, that is, they are photosynthetic. They can synthesize their own food. They are autotrophs, and they can occur in a variety of colors, such as they can occur in yellow, green, brown, blue, or red color. So these dinoflagellates, they have pigments in them. So the pigment can be either yellow pigment, green pigment, brown pigment, blue or red pigment. Depending on what pigment is present in the dinoflagellates, there are different types of dinoflagellates. And not just that, actually, if I told you dinoflagellates are marine. If lot of dinoflagellates are present in the sea, it will make the sea red color or blue color depending on the pigment that is present in them. So yellow, green, brown, blue or red color, they have pigments. Depending on, they can be of different colors depending on the pigments that are present in the cells. And the cells, they are covered by a cell wall, and the cell wall is made up of cellulose. And in the previous session, you have seen the cell wall. Protistans, they have a cellulose-rich cell wall, and the cellulose plates are present on the outer surface. That is why they are hard. They form hard plates because of the presence of the cellulose. So the cell wall of dinoflagellates, they are very stiff and hard because they are made up of cellulose plates. That is, the cell wall is accumulated with cellulose. That is why it is very stiff, because it is made up of cellulose. And they are called dinoflagellates. You should remember, whenever the term dinoflagellate comes, it means they have two flagella. So one flagella lies longitudinally and the other flagella lies transverse. So if this is the dinoflagellate, say for example, one flagella will lie like this and the other will lie transverse like this. So it has two flagella. One will lie transverse and the other will lie longitudinally. So two flagella, one lies longitudinally and the other, it lies transversely in a furrow. So I told you they are made up of wall plates. So if this is the dinoflagellate, they are made up of two wall plates. So they are made up of two wall plates. So in between the wall plate, there is a small furrow here, or a gap or an opening. So from there, one flagella is present and the other is present transversely. One is longitudinally and the other is present transversely. So the two flagella, one lies longitudinally and the other transversely in a furrow between the wall plates. So these are the wall plates. So these are the wall plates. So in between these wall plates, there is a small opening or a furrow. small depression is there. There is a small depression or a furrow from which the two flagella they arise. One longitudinal flagella and one more transverse flagella. Two flagellas will arise. And the best example is Gynologs. So Gynologs, it is one of the best example for dinoflagellate, and this Gynologs is red in color. Therefore, it makes the oceans. So wherever in the ocean there is a large amount of Gynologs dinoflagellates present, these Gynologs, they have red color pigment in them. That is why it makes the ocean red in color. So we shall see about Gynologs. So Gynologs, they divide, they reproduce by rapid multiplication again here, and therefore they make the sea appear red. So you can see here, they make the sea, you can see the seashore here, it appears red because of this Gynologs. It contains red pigment in them. So when they divide or reproduce or multiply very quickly, when a large number of Gynologs is formed, the entire sea will look as if it is red in color because these Gynologs have red pigments in them. Just like that, there are some dinoflagellates which have blue pigment in them. You might have seen in National Geography channels and all, wherein when the waves come, no, the waves will look as if somebody has lit bulb blue color lights in it. It is called bioluminescent. So they look blue in color. Some dinoflagellates, if they are blue in color, they make the ocean waves appear blue in color. It also tells that they are marine in habitat. And apart from that, they produce certain toxins. So this is one thing. They make the oceans look beautiful because of the colorations present in them. But one more thing is, they as the number of Gynologs or dinoflagellates increase in number, they start to release certain toxins, and these toxin accumulation, actually it is harmful to the marine animals, especially the fishes, and they also can kill. They are poisonous or they are toxic to the fishes and they can kill the fishes. So this is about the dinoflagellates. Next, moving on to Euglenoids. So Euglenoids is also a kind of Protista, which is a freshwater organism. Especially they are found in stagnant water, that is non-flowing water, the water which are still, which do not flow. So in such stagnant waters, these are present. Fresh water, they are present in fresh water, especially in the stagnant waters. And they have a pellicle instead of a cell wall. So dinoflagellates, they had a cell wall which was made up of cellulose. But these euglenoids, they do not have a cell wall, but instead of a cell wall, they have something called as pellicle. They have pellicles instead of cell walls. They have pellicle. So they have a pellicle instead of a cell wall, and this pellicle, it is rich in proteins. It is rich in protein, and therefore, since it is rich in protein, it makes the body of the euglena. So the best example for euglenoids is euglena. Since euglena has a protein rich layer, which is nothing but pellicle, they are very, very flexible in nature. They can bend however forms or shapes they want because they are flexible. And why they are flexible? Because the layer is rich in protein, and they are made up of two flagella. They are made up of two flagella. You can see here, one and the other one. They are made up of two flagellas. They are made up of two flagellas, a short and a long one, a short flagella and a long one. And they are photosynthetic in the presence of sunlight. So they can be photosynthetic and they can also be heterotrophic. So whenever there is presence of sunlight, they can be phototrophic. So these organisms, they have a flagellum and they have one more small flagellum, and they are photosynthetic in the presence of sunlight. There is an organ called as photoreceptor. Here, can you see here this particular organ? It is called as photoreceptor. Photo means light, receptor to receive. So because of the presence, whenever there is sunlight, these photoreceptors, they receive the sunlight and they act as photosynthetic. They make the organism autotrophic, that is to synthesize their own food. And apart from that, they are autotrophic, they are photosynthetic because of the presence of chloroplast, and they also have these euglenas or euglenoids. They also have various cell organelles such as the mitochondria, the nucleus, then they have the Golgi apparatus, they have the contractile vacuole, the endoplasmic reticulum, and they also have carbohydrates in the form of reserved food material. So carbohydrates, whenever they require a source of energy, they can utilize the carbohydrates. So this is about euglena. So whenever they are deprived of sunlight, so I told you they are photosynthetic or they are autotrophic only in the presence of sunlight. Sometimes maybe deep in the stagnant waters, because of algal bloom, what happens, sunlight may not be able to penetrate. So whenever they do not get the sunlight or whenever they are deprived of sunlight, they act like heterotrophs. That is, they start feeding on other smaller organisms that are present in the stagnant water. They start predating or feeding on other smaller organisms that are present in water. Therefore, one of the best characteristics of euglenoids under the kingdom Protista is that they can be autotrophs as well as they can be heterotrophs depending on the presence and the absence of the sunlight. So this is about euglenoids. So next, moving on to one more kind of Protista member, that is slime moulds. So slime moulds are saprophytic, means they usually feed on the dead organic matter. So wherever there is a dead wood or something, it will start growing there, these slime moulds. And the body of these, they actually moves along the decaying twigs, and that is, how do they feed is, they move along on the decaying twigs and they engulf the decaying dead matter. That is how they are saprophytes. That is how they feed on the dead organic matter that is present in the terrestrial earth. So that is what happens. And during favorable condition, they form a group or an aggregation. So during the favorable condition, they form a group called as Plasmodium. That particular group is called. So these slime moulds, during during favorable condition, they form a aggregation or they form a group, and that particular group is called as Plasmodium. So this Plasmodium, they grow or they spread in groups. So you can see here, they are usually individuals, but whenever favorable condition is there, they can occur in groups. They can occur in groups and they can spread on the entire dead organic matter. So that is why they are the best saprophytes. And during unfavorable condition, they differentiate. Differentiating means this body of the organism gets converted into a different forms. They develop a new form. That is nothing but they differentiate and they form fruiting bodies bearing spores at the tip. So what happens here is, during favorable condition, they form groups and then they spread. But during unfavorable conditions, they develop spores. So you can see spores here. So they develop fruiting bodies. You can see here everywhere fruiting bodies are there. So they develop fruiting bodies like this. So that is during the unfavorable condition, the body of the slime moulds, they differentiate and they form fruiting body like structures at the tip. And these fruiting bodies, they have spores inside them, which actually helps in reproduction. That is asexual method of reproduction. So the spores, whenever wind is there, the matured spores, they will disperse, and wherever they disperse and fall on, again there the saprophytes, that is this particular slime mould, it will start to grow. So the spores, they get dispersed by the air currents, and also one of the important characteristic of the slime mould is that they possess true walls. Hence they have a true wall. They have wall layers, cell wall covering layers. That is why they can survive in any extreme condition. So they possess true walls, hence they are extremely resistant to, maybe sometimes the soil may become acidic, sometimes it may become basic. So when the acidity increases, it is difficult for the organisms to survive. So in such conditions, what happens, in spite of any kind of environmental condition, because they have a very rigid true cell walls being present. They are extremely resistant to any kind of environmental condition, and therefore they can survive for many years. These slime molds, they can survive for many years, just like the diatoms as we had discussed. So this is about the protists. So there is one more form of protists, that is amoeboid protozoans. So amoeboid protozoans, they live in fresh water, that is ponds, rivers, lakes, etc. They can also live in sea water, and they can also be terrestrial, but that is moist soil. It means that they always require water. These amoeboid protozoans, they always require water, and that is why they also live in soil, but the soil should be moist, that is laden with water. And these amoeba, they move and capture their prey. So you might have seen how the amoebas, like when you observe under the microscope, no, they can move. They move and they capture their prey. So they capture their prey by putting the pseudopodia on the prey. So this is the pseudopodia. So you can see here, this is the pseudopodia. So they cover the organism with the pseudopodia. So if this is the organism, or if this is the material which the amoeba wants to eat, it will just move, slide towards that material, and it covers its pseudopodia or its body on the material, therefore engulfing or eating the organism as such. So they move and capture their prey by putting the pseudopodia, which is nothing but also called as pseudo, means false, podia means foot. So that is why it is called as false foot. So it looks like foot. Why it is called false foot? Because it is not foot, but in spite of that, it helps the amoeba to crawl and reach its particular destination. That is why it is called as pseudopodia or false feet. And there are also marine forms. I told you they can also live in marine environment, that is seas, oceans. So these marine forms, they have silica shells on their surface. So they have silica, just like diatoms. How diatoms have the silica on their surface, on their cell wall, these also have silica shells on their surface. And the best example is entamoebas. And this entamoeba, they are usually parasites, and they cause diseases in human beings as well as the animals. So next, moving to flagellated protozoans. So these flagellated protozoans, they are free living. They can be free living. That is, they can live on their own without depending on any of the other organism, or they can be parasitic. Parasitic means they depend on other organism for their survival, and whichever organism they depend on, no, they will completely kill or cause the death of that particular organism. That is why it is called as parasitic. And they have flagella. That means they are motile. They have flagella which help them in movement, and they are parasitic. As I told you, they are parasitic. One of the important disease that is caused by flagellated protozoans is the sleeping sickness. So what these flagellated protozoans will do is they will start attacking the red blood cells. When they infect the red blood cells, it will cause a disease called as sleeping sickness in human beings. The best example is trypanosoma. So trypanosoma, usually it is the best example for a flagellated protozoan, and it actually infects the red blood cells and it causes sleeping sickness, which is a disease. Next is celiated protozoans. So I told you protozoans, they can either have flagella or cilia. So fast we had studied the different types of protozoans which had flagella. We did not come across any of the protozoan which had a cilia. So now we'll talk about the celiated protozoans. Those protozoans which have the locomotory organ in them called as the cilia, which help in movement, which make them motile. So celiated protozoans, they can either be aquatic, they usually live in water, water is their habitat, and they actively move because they have cilia, which you can see here, they have cilia. This is the hair-like covering that I was talking about. They have cilia, and this cilia is like having hundreds to thousands of feet. They will actually help in the movement. They can actively move because of the presence of thousands of cilia. And they have a cavity which is also called as gullet, and this particular cavity opens outside towards the cell wall. So you can see here they have a cavity. So this cavity, it is also called as gullet. It is also called as gullet. They have a cavity and this opens externally towards the cell wall, and they have a coordinated movement. Why coordinated movement? Can you see? Imagine the cilia here moving backwards and front at a stretch. So it will give a coordinated movement. So it has a movement like this. So because of this movement, one side of the cilia, they will move up towards one direction, the other will move towards the other direction. This will cause movement of the organism. So the coordinated movement of the rows of cilia causes the water-laden food. So when the coordinated movement occurs, so it is like a brush. When it is brushing, what happens is the water, whatever water is there, it will enter into the gullet. So it will enter into the opening or the cavity or the gullet. At that time, whatever organism is present in that particular water, it will enter into the paramecium or it will enter into the celiated protozoan. That is how they engulf their food. So what happens here? The coordinated movement of the cilia causes the water laden with food to be stirred into the gullet. So when there is movement of the cilia, imagine when there is movement, it will push the water into, so if this is the gullet, imagine the cilia is moving like this here. It will drag the water. It will push the water into the gullet. So when it enters into the gullet, whatever food material is there in the water, it will enter into the organism. That is how they feed. So this is how they feed. So the best example for celiated protozoan is the paramecium. Paramecium is one of the best examples. So cilia and cavity or gullet is the main organ that is present in this particular organism. So they are heterotrophs. They depend on other organisms for their food. So the food enters through the gullet or the cavity. So next we'll talk about one more Protista group, that is the sporozoans. So sporozoans, as the name suggests, they are spore-like. So they produce infectious spore-like structures. So they have in their life cycle, they have a particular stage wherein they produce the spore, and these spores, they actually cause infection. So they have an infectious spore-like stage in their life cycle. The best example is the Plasmodium malarial parasite. Do you know how does malaria spread? Spreads through the mosquito bite. Will all the mosquito that bite you give you a malaria? No. Inside the gut of the mosquito, this sporozoan will be present. The best example is Plasmodium. So this Plasmodium sporozoan will be present. So why it is a sporozoan? Because in the life cycle of the Plasmodium, they'll enter into a spore bearing stage. So the spores will be present inside the mosquito, and that spores are the ones which causes the infection. So when that mosquito carrying in its gut or the stomach, carrying the spores of the sporozoans, that is the Plasmodium, when it goes and bites a person, it will transfer the spores into that particular person. Therefore, those spores will cause malaria disease in the human being. That is why I told you they have a infectious spore-like stage. So, which usually that spore-like stage is the one which causes infections or diseases in human beings. The best example is Plasmodium, which is a malarial parasite, and it causes malaria in human beings. So that Plasmodium in the form of spores, it will be in the, how will it enter into the mosquito? Maybe through water or something. It will enter into the gut of the mosquito. So once the spores are present in the gut of the mosquito, when the mosquito sits on the human beings to bite them, it will travel to the bloodstreams of the humans, and therefore it will cause the infection, malaria. So that is how they spread. So this is about, you can see here, the malarial parasite, that is the Plasmodium being present. So this is about the malarial parasite, which is nothing but a sporozoan, and the best example is Plasmodium. So this was about the kingdom Protista. So I hope you have understood the session well. So we had studied about the second kingdom of Whitaker's classification, that is Protista. So in the next coming session, we shall discuss about a vast kingdom, that is the kingdom fungi. So there is a lot and interesting facts to learn in that. So we shall meet in the next coming session. Thank you.